welcome to RCA Radio, a podcast where we cover the latest news and challenges in regulatory, compliance, and quality assurance facing the life science industries. I'm your host, Brandon Miller. In this episode of RCA Radio, we'll be reviewing the recent draft guidance document, Chemical Analysis for Biocompatibility Assessment of Medical Devices, and the implications this guidance will have on the industry. Today, I'm joined by Nelson Labs Director of E&L, Raymond Colton, as well as board-certified toxicologist, Dr. Sarah Campbell, also from Nelson Labs. Welcome, Ray and Sarah.
Hello, Brandon.
Hello, Brandon.
Thank you both for joining me today. As I said before, we're going to talk about the recent draft guidance document that the FDA is working on and the impacts it will have on the industry moving forward. So to start off, can you guys both explain what this new guidance is about and why it's important to our industry?
Sure, I'll take that, Brandon. Thank you. First of all, I'd like to say that I've been in the E&L testing market for about 25 years. In those 25 years, I've never been in a situation where the FDA requests and guidances are changing as fast as they are right now under CDRH. So this is a fascinating time to be in this field. It's very significant for this industry because the requests that are being asked of the labs and of the medical device companies have evolved so that there's much more scrutiny and detail that is required in order to have a positive chemical characterization test that leads to a successful TRA. Sarah,
Thanks.
You want to go?
Yeah, I was just going to say, I have been an analytical chemist for 12 years. I've been a toxicologist now for a couple decades. And yeah, this is a very challenging time. And since the ISO 10993 Part 18 standard came out in 2005, it really was the second version that came out in 2020 that left a lot of subjective language and a lot of CROs to interpret what the testing requirement should be. And I would say that this new FDA guidance is really putting that subjectiveness from the Part 18 standard into more an objective viewpoints of what their expectations are. And my hope for that is that it really can lead to some better consistencies. And, a lot of these best practices that are in the document were implemented along the way at Nelson Labs already.
That's great news to hear that you guys are already starting to implement some of these best practices. So what are the potential impacts that this guidance can have on device manufacturers?
Certainly from a testing standpoint, you have to plan further in advance. There's been parts of the testing that now just plain take longer. What used to be 24-hour extractions, it's recommended to be 72-hour extractions. The number of tests that are required to reach exhaustivity may go up with these new requirements. So you need to add more time and probably, honestly, more of a budget. But there are some very positive aspects because I think these new requests with a consistency, as Sarah mentioned, will lead to a better result with your TRA.
Yeah, and I'd also like just to say, the FDA in this documentation really is allowing for justifications when testing gets challenging. If you have a simple implant device, this may not impact you as much. But if you have really complex device, a biodegradable device, or, something that's a reusable device, you really have a lot of justifications that the FDA is now stating that they allow. So one of the things that I like to see was that they're going to allow 2 solvents only instead of 3 solvents. That's cost savings for our sponsors. And all we need to do is justify that the polarity of the solvents that we are using match those biological tissues or fluids that the device is going to be in contact with. So that's just one of the good things that can come from this guidance. And I think what it allows for is if something happens that is not necessarily standard or the ISO 10993 Part 18 document doesn't cover in detail, they've now put out what their expectations are. And so what my hope is that sponsors receive, dare I say it, no deficiencies, but really a limited number of deficiencies that are really just clarifying questions.
So with the FDA and how they're putting out these different clarifications, how rigid are they going to be with it? Is there potential for them to grant any leeway depending on the type of product?
My experience with that is, there's certain aspects of testing that are going to be non-negotiable. Your instruments have to have a sensitivity that detects low enough in order to make a meaningful TRA. That's good science. That makes sense. In order to achieve that, though, the FDA, in our experiences, has shown willingness to bend on other areas in order to achieve the most important results, like having the sensitivity. An example of this would be a case where a test article absorbed a lot of a solvent, so we could not achieve the surface area to volume ratio that they recommended. It was okay for them in that case to, let's say, double the volume of that solvent in order to have enough to recover and do testing with. So it's not unreasonable. That's still good science. We still have AET that's well above the LOQ, and that was a very productive interaction with the FDA.
Yeah, so, and I also think with this new guidance, we'll have less guessing of what the FDA wants, and we'll have more allowance for these scientific justifications when things are challenging. And so I really do think that the FDA has done a really good job of writing this draft document. We plan to comment on this draft document, and we also plan to be on site at the FDA workshop on the ASCA certification for chemical characterization reporting. That a lot of the stuff that they've now put in writing in this draft document, I've seen and worked through over the last decade, helping sponsors with submissions. Not every device can just be put through ISO 10993 Part 18 testing and have it go as planned. And so I'm seeing that the FDA recognizes that. And I've also worked with many FDA reviewers where we've allowed for some scientific justifications to waiver in order to get chemical characterization completed on a device.
That's great to hear that Nelson Labs and you both are already starting to implement some of these best practices while testing. And it's also amazing to hear that you guys are actively working on comments for this draft guidance and attending the meeting that will go over this guidance. With that being said, what are some of those best practices that you have already implemented at Nelson Labs?
I've got a great example of that, Brandon. Thank you for that question. And I want to stress that it's really important, the progress that we've made in this area, it's really important that the chemistry group and the toxic group work very closely together. And that's something we really do a lot at Nelson Labs. So, these projects are coming to the chemistry group first as we're designing how to do the test and what tests should be done. But we often have the tox group join us for phone calls with the potential sponsors to make sure that we're going to have a clear pathway to a successful TRA. One change in the draft guidance compared to how we did it, say, a year ago, is that we used to determine the exhaustive time period, using NVR, and that might be 3 24-hour extractions or 4 24-hour extractions. And then we do the chemical characterization based on one extraction from the entire time. So it might be a total of 72 hours. Now it's very clear that we are to do the extractions individually. So if we say it's going to be 3 extractions, we perform all 3 extractions and do the full chemical characterization on all 3 extractions in order to show the concentration of the different extractables go down over time. Now, this does add cost for sure. If you're going to be doing more testing, it will add costs. However, it really has the potential to lead to a much easier TRA once the tox group takes that data. And Sarah, why don't you explain what we've done at Nelson?
Yeah, thank you, Raymond. So one of the things that we can do with those 3-point data points is we can take those first data points and set a different threshold of toxicological concern. Those would be essential time points within acute, subacute type periods for patients. And so we can reference toxicological thresholds for those earlier time points. And we know that those are generally always higher. So you can have a higher exposure on those first earlier time points than you can at that later time point, which we have to assume could be chronic. So that is the expectation. So we can apply some toxicological tools. And in doing so, what that can do is it can allow us to just solve any potential toxicological hazard with the data already collected with the extractables, and therefore potentially not need to do any follow-up leachable or simulated use kinetic studies, which could extend the time and, of course, extend costs to our sponsors. So it's a really great addition. Another best practice that I think is really important, and I think Nelson Labs is pretty exclusive to this, is about a decade ago, I worked with the expert chemist to develop our ICP-MS method. And what we did at that time when we were developing that method is that we look not only at what the ICH Q3D elemental impurity stated as your permitted daily exposures. Those are for pharmaceuticals. We have really unique contacts in medical devices, really unique exposure routes, really unique scenarios that are not replicated in pharmaceuticals exposure. So what we did at that time is I worked with the chemists at that time, and I myself had developed several mass spec methods in the past. So we worked with what should our mass spec method for elements be. And what we did is we did a really conservative, great approach is we looked at what would be the most toxic element, and we even considered the most toxic valence state if it were that. Now, again, ICP-MS doesn't give you that valence state information, but we assumed that it could be this most toxic valence state. And we went to an exposure limit that would be safe for a preterm neonate. And we set our limits based off of that. And I don't know if any other lab can say that they've done that. So we now have, with any analytical run for our elements, protection for those very sensitive preterm neonates. And that's one of the best practices that's mentioned in this FDA guidance that we implemented almost a decade ago.
It's great to hear that Nelson Labs and you both have been working with these best practices before they have been even published as a draft guidance that will help with implementation once this becomes a published guidance. And with that said, we are running low on time here. So I want to pass the ball off to you guys and get your final thoughts and maybe a key takeaway for the listener.
Sure. Thank you, Brandon. I think my key takeaway is that companies should be planning this in advance and maybe starting a bit earlier. But also, in order to do the justifications that Sarah mentions, it's important that you really understand your process, your manufacturing process. The more you have knowledge about how you manufacture so that you can justify the consistency of your product, minimize the potential that you have to test multiple lots, and help the chemistry laboratory understand what potential extractables could be there, will speed the process along and lead to a more successful application.
And I would just follow up with saying, plan for the unexpected. You may have done chemical characterization in the past. If it was years ago, know that this new draft guidance is potentially going to add some cost and increase your timeline. And really one thing that I want to state is that a pre-submission can really help work these details out. Now, since there's going to be this guidance out there, I think we've got a lot of things that we were discussing in pre-subs now written down on paper, and I think that we can follow that. But I still think for very unique devices, let's still get those pre-submissions going. Nelson Labs can join you on those pre-submissions and get those details worked out with the FDA about what their expectations are for your unique device. And we also can bring those justifications to that pre-sub so the FDA can see them, review them beforehand, and be able to discuss them with you on a path forward.
It sounds to me like preparation is key, and contacting the right experts will go a long way. And with that being said, I'd like to thank you both for taking the time to go over this guidance and the impacts it will have, and how you and Nelson Labs are both working to assist the FDA to get this guidance across the finish line. And I also want to thank our listeners for tuning into this episode of RCA Radio. Be sure to subscribe to be the first to know when we upload the next episode. Thanks again, and have a great day.